So this is a summary data. Actually, it's on your also in your textbook, 107. So if we look at different regions, then we can compare all the differences. And of course, given all the knowledge we have with the DNA sequence and amino acid sequence, then we can compare only to a certain part of the gene. For example, we can compare the coding sequence, and we also compare the non-coding sequence. It's always very difficult to define the functional domain of the non-coding sequence. Even today, we have a lot of genetic and genomic tools, but we still cannot decode every single base or annotate every single base in the human genome, or even in a C. elegans genome, which is much smaller. Okay, so in the non-coding sequence, our difficulty is to separate the functional region and non-functional region. Just want to remind you, this is a diagram. Try to look at the sequence at surrounding a gene. And we already, from molecular biology, we already know there are certain signals or signatures at the DNA sequence level. It's important for gene function. For example, you need splice sites for splice the introns. You need promoter sequences for transcription. And you also need some 3' UTR for to determine where to stop. And the 3' UTR sometimes also is important for direct the RNA molecule from the nucleus to the cell, and sometimes they have to wait for a while before translation. So all these molecular properties affect the functional constraint or cause the functional constraint of each region. So this is an early comparison, and we can separate the regions and try to compare the differences. So one obvious thing appear here is they all appear quite differently. So from the 5' franking, so this is the 5' region of the gene, but not including the 5' UTR. And to be sort of fair, we use the 4' degenerate site of the coding sequence. So that's the one we know they don't change the amino acid. So they can, regardless the codal position, they won't change the amino acid sequences. Then, of course, intron is another or a big chunk of non-coding sequence sitting within a gene, and also you can do 3' UTR and flanking region. And the last one is the pseudogenes. Pseudogene is the one supposedly every change at the locus doesn't really have any consequence in terms organism's fitness. So with this comparison, we'll see, well, our expectation was they should be very similar. But you do see differences, especially at 5' UTR, 5' franking. So then the next question is why these two regions are special in our coding sequence gene, our protein coding genes. Okay, this is another exercise. When you see differences, we ask why they are different. When you do not observe differences, then we ask why they are the same. So the observed mutation rate at the 5' franking and 5' UTR are much lower than the rest of the gene because of the functional constraint, right? So in this pattern, you can think of the possible reasons. But as I say in the first page of this chapter, I already told you, right? Mutation rate, fixation rate, and why they're different. So here, if we look at the 5' region and all the rest, we can separate these two large groups first. Then you observe here has more constraint for their substitution rate. The observed rates are lower than the others. So when I say I sort of try to emphasize the observed rate, right? So the rates can be detected by us. And those are the ones survived from all the random process, population fluctuation, and natural selection. So most of them are that. And you also learn that from your genetics, biology, and molecular biology. Gene regulation often is at the 5' of the gene. It doesn't mean the 3' is not important. So sometimes cis-regulatory element, when we say cis-regulatory element, is like at the surrounding region, very similar region, or on the same chromosome. Some people use that too. But cis-regulatory elements are often located at the 5' of the gene. They are cis-regulatory elements in the introns, and they are important sequences at introns, right? And they are important sequences at the 3' of the gene. But the large difference, the large amount of the information is at the 5'. So that's why the dramatic differences in this table is the difference between the 5' region, or say cis-regulatory region, and the rest of the regions. And you do also observe a little bit, the pseudogenes are the one evolved most rapidly in this case. We usually use pseudogene as a neutral marker because it has no functional constraint. So that means all the mutations there do not have any fitness consequence. So they don't code for protein, they do nothing, they just sit in the genome. It's just time is not long enough, so we still be able to recognize as, say, the pseudogene of the hemoglobin, but has no function in our cell at all. So this is being used as a neutral comparison. And if you want to discuss a little bit between the second class, or the A point something, 8.0 class, to the pseudogene, and that reflect what I say, the sequence at these positions are still important in terms of the gene function. So not every single site can freely evolve to all the others. So when we start from chapter 3, we say, oh, every site A can change to C, G, and T. Yes, that's by mutation, right? We're purely talking about mutation could occur at any position, to any direction. That's purely start from only mutation. But now we want to look at chapter 4, we're looking at the real data, the data we observed, not only contribute by mutation, but also by other evolutionary mechanisms. So that's why we're so interested in observe the pattern from our samples. So this is something I want to emphasize.